Okay, so we are finishing off chapter 4 now on neuronal physiology. And today, this lecture, we're focusing on neuromuscular synapses. And then we'll also look at some synaptic integration, okay? So just to start off with, remember we've been looking at neuron to neuron synapses, okay? So neuron to neuron connections. Now we look at a different kind of, the second major type of connection in animal body, which is a connection between a neuron, a nerve cell, and a muscle cell, okay, which is known as a muscle, skeletal muscle fiber. So, the neuromuscular junction is the official term for the synapse that we have between our motor neuron. So remember, that's a neuron that connects with muscles, and it generally affects an action, a contraction in a muscle, or a relaxation, but it connects with muscles, okay? So this is a motor neuron, okay? So the motor neuron, so the axon of a motor neuron, okay, as it approaches a muscle, it divides into many terminal branches, and it loses its myelin sheath, okay? So each of these terminal branches innervates, which is the term which means basically to supply with nerves, okay? So when I say innervate, I'm talking about a nervous connection with another cell, okay? So each of these branches, these terminal branches of a motor neuron, it innervates, each branch innervates with only one muscle cell, okay, known as a muscle fiber. So therefore, each muscle cell has only one of these neuromuscular junctions, okay? So it's a one-on-one thing. So a single muscle cell, which we call a muscle fiber, is a long and cylindrical cell, okay? So it's long and cylindrical in shape. And we'll look at a picture of that coming up next. So if we look closer at the structure of a motor neuron, the ending of the motor neuron, the ending of the axon of a motor neuron is called the terminal button, okay? And it's basically where the motor neuron, which is a thin axon initially, it spreads out into a wide sort of round button-like thing. Okay, so that's the terminal button, is the ending of the motor neuron, and it fits into a little groove or depression in the muscle cell, okay? So in the membrane of the muscle cell. And this little groove is known as the motor end plate, okay? So this is the part of the receiving muscle cell where the terminal button of the motor neuron connects, okay? Now we have a substance called acetylcholine, we refer to as ACH. And this is our neurotransmitter, okay? So as with our neuron to neuron connections, we have a neurotransmitter governing neuromuscular synapses, okay? And in all cases, it's acetylcholine. That is the neurotransmitter that is stored in the vesicles of this terminal button and is released and crosses the synaptic cleft and causes muscle contraction in the receiving muscle cell, okay? So just a micrograph here of showing the so just a muscle picture of muscle fibers, these long, thin muscle fibers. And here you can see the motor neuron, okay, has these thin axon terminals, okay, and each terminal innervates or connects with a muscle fiber via one of these terminal buttons that you see here, okay. So this is what it kind of looks like in real life. And each of these little terminal buttons would cause contraction of a muscle fiber, okay? So if we look at the process of a neuromuscular synapse and how it all happens, we start off with an action potential, okay? As with the neuron-to-neuron synapse, we have an action potential that is flowing down the axon of the neuron, in this case, a motor neuron, and it arrives at the terminal, so the button of the presynaptic neuron, okay? And just as with the neuron-to-neuron process, very similar. It opens, the action potential causes the opening of these voltage-gated calcium channels that are in the terminal button, okay? And this allows a large influx of calcium ions to enter. Now, this is pretty much exactly the same as with the neuron-to-neuron process, okay? So the neuron-to-neuron synapse. So as with the neuron-to-neuron synapse, our neuromuscular synapse causes at this point, the action potential causes the release of a neurotransmitter by exocytosis of the vesicles, those little bubbles that hold the neurotransmitter inside the ending of the neuron. And in this case, it is our acetylcholine, okay? So the ACH molecules are released out of the presynaptic neuron, and they cross that little synaptic cleft, as would be very similar to the neuron-to-neuron connection. But in this case, these ACH molecules bind to protein receptors called cholinergic receptors, okay? So these are a particular type of receptor that are receptive only to acetylcholine, and they are situated on the motor end plate, okay, in the little groove in the receiving muscle cell, okay. So the acetylcholine, it binds to these receptors, okay, and opens chemically gated ion channels in the motor end plate, which leads to depolarization, okay, which is very similar to the process for neuron-to-neuron synapses. In this case, the depolarization that results is called end plate potential, okay? So it's happening in the motor end plate. So we call this EPP. Now, once this end plate potential happens, it basically can in turn cause an action potential, which spreads to the rest of the muscle cell in both directions, actually, via contiguous conduction, which we've already talked about. So there's no myelin sheath around a muscle cell, remember, around a muscle fiber. So action potential is moved by contiguous conduction. The end result of this whole neuromuscular synapse process is to have is for the action potential to initiate contraction of the muscle fiber, okay? So this contraction of the muscle fiber happens en masse. There's a whole lot of these fibers contracting. And when they contract all together, you would be able to do things like move your fingers, hold on to things and basically move your body, okay? That's sort of the end goal of this whole process. So just to note that the EPP, the end plate potential, it brings about a graded potential, okay? So this is because the motor end plate region itself does not have a threshold potential, okay? So the action potential cannot happen in the motor end plate itself. So you have initially you have a graded potential that's produced by the chemical, this chemical synapse, okay? However, this graded potential, this EPP, it sets off an action potential in the adjacent cell membrane, okay, which can then spread along the muscle fiber. So that's kind of how it works. Let's look at a picture of this. According to your textbook, we have a 9-step, an 8-step process, but I'll add in a 9th step here. Step 1, we essentially have your action potential in the motor neuron is propagated to the axon terminal, okay? So this terminal button. Now remember, the myelin sheath covers most of the axon of the motor neuron, but then you don't have the myelin sheath disappears when you get right to the end of this axon, right to the terminal, because obviously the myelin would block any flow of ions, okay, out of the synapse. So basically your action potential arrives at this terminal button, okay? This local action potential, it triggers the opening of voltage-gated calcium channels, okay? And this allows the subsequent entry of calcium ions into the terminal button, okay? So you've got this large influx of calcium ion. Now, this influx of calcium ions triggers the release of the acetylcholine molecules, these little tiny molecules, by exocytosis of these vesicles, right? So these little bubbles that hold the substance, they leave the terminal button, okay? So this releases a whole lot of this neurotransmitter substance, these molecules, which diffuse across the space, separating the nerve, separating this neuron and muscle cell. And the substance binds with receptor channels that are specific for this receptor, right? So it binds with these channels, and this is in the what's known as the motor end plate part of the receiving muscle fiber, okay? So this binding over here of neurotransmitter acetylcholine with your receptor channels brings about opening of these non-specific, what's known as non-specific cation channels, okay? So they're not specific to sodium or potassium. Either of these molecules can flow through these channels, okay? Now, this binding, what I want to emphasize is that it leads to a relatively large influx of sodium ions into the muscle cell, okay? And only a relatively small movement of potassium out of these channels into the into this synaptic cleft, okay? So this is because of the electrochemical gradients that we find existing between the inside and the outside of the muscle cell membrane, okay? So sodium rushes in, because remember, you've generally got a more negative charge at resting potential inside an animal cell, and a more positive charge outside. So this concentration gradient governs a large influx of sodium. And there's also electrical gradient, because positive charges are attracted to negative charges, okay? Potassium, some potassium flows out at this point also along its concentration gradient. But it is not governed by, it is only flowing along a concentration gradient, not an electrical gradient at this point, because remember, potassium plus ions, the general going against the electrical flow of a resting potential membrane, okay? Don't worry too much about the details there, but just understand that at this point, sodium rushes in and some potassium moves out, okay? So the result of this is what's called end plate potential, okay? So EPP. So this EPP is a graded potential, okay? And it now changes it causes a small depolarization, basically. So a small change in polarity towards a more positive state inside the muscle cell, right? But this graded potential is not at threshold, but it causes local current flow, because remember, the charges now move to the adjacent part of the cell membrane from positive to negative, right? So you get this movement of what's called passive diffusion. So the movement of the graded potential to the adjacent part of the muscle cell. And then the adjacent part of the muscle cell is loaded with sodium, voltage-gated sodium channels, okay? And these are sensitive now to subtle or small depolarization. So now, you know, your graded potential, your small depolarization arrives at an adjacent part of the membrane, and it causes an action potential now, because it triggers a bunch of sodium voltage-gated sodium channels to open, and you have an action potential sets off, and this now can carry on along the muscle fiber, okay? And this happens in both directions. So you should understand that these neuromuscular, these terminal buttons, so the axon connection, sorry, let me say that more clearly. The motor neuron connection with a muscle fiber generally happens toward the middle of a muscle fiber cell, okay? It just works out that if it connects in the middle and causes an action potential to go in both directions, right? So that way and that way. If this happens, it turns out that this is an efficient way of causing contraction of the muscle cell as a whole, okay? So this movement in both directions causes contraction of this muscle fiber. I've added in there's a step 9 that is talked about in the textbook that is not in the diagram, but basically once this whole process has happened, you need to get rid of the neurotransmitter molecules. They can't hang around here carrying on. If they were to carry on they would carry on connecting and fusing with these receptor molecules. They would cause continuous action potentials to be set off in the receiving muscle cell. So we have a substance called acetylcholinesterase. Good luck remembering that name. So this acetylcholinesterase is an enzyme, and it's located in this motor end plate region. Okay, so here's your acetylcholinesterase. And this substance, it basically destroys the acetylcholine molecules, okay, and allows one to reset this process, basically. Okay, so I just wanted to show you a quick animation of this whole process. See if it helps a bit, and might give you a couple of minutes to just clear your head a little bit. So just give me a moment while I set this up. Okay. All right. Let's have the...
A neuromuscular junction is a chemical synapse between the axon endings of a motor neuron and a muscle cell. A narrow synaptic cleft separates the presynaptic cell, the motor neuron, from the postsynaptic cell, the muscle cell. The presynaptic cell contains vesicles filled with neurotransmitter. The postsynaptic cell has receptors for these vesicles. Here's a closer look at the postsynaptic membrane. The neurotransmitter can bind to a receptor site in a gated channel protein. Binding of neurotransmitter to the receptor site causes the gated channel to open. Ions flow into the postsynaptic cell and trigger an action potential at the membrane site.
Okay, I'm back. That was a very short little animation, but just thought that it might help. Okay, so let's just bring back the PowerPoint quickly. Now, okay. So I'm trying to catch my line of thought there. Okay, I know what I'm going to say now. So unlike with synaptic transmission, when you have a neuron-to-neuron connection, as we talked about earlier, the magnitude of a single EPP, or a single end plate potential, is normally sufficient. Okay, the magnitude of these EPPs is normally sufficient to cause an action potential in the muscle cell. Okay, so therefore, basically, one action potential in a neuron triggers one action potential in a muscle cell that it innervates. Okay. Just wanted to highlight that. The EPP that results in a muscle fiber, so this graded potential, is much larger than the EPSP that is generated in a neuron-to-neuron connection. So remember, EPSP is your excitatory postsynaptic potential. Okay, so that was the graded potential that we saw happening in the neuron-to-neuron synapse. Okay, so the reason that the EPP in a muscle fiber is much larger, so the reason that this graded potential is much larger than for a neuron-to-neuron connection, there's a couple of reasons behind this, and one of them is that, if you think about a neuron-to-neuron connection, it's just one neuron connecting onto one neuron. But here with a neuromuscular synapse, we have multiple branched ends of one motor neuron connecting with with various motor end plates on a muscle fiber. So you've actually got you've got a whole bunch of these connections, right? Because the axons, the terminating point of the motor axon, forms all these little branches. Okay, so that's one reason. Another reason is the neurotransmitter. There's basically a whole lot more of these acetylcholine neurotransmitter molecules that are released at each terminal button. Okay, so if you have more neurotransmitter molecules, you are likely to be able to have more receptors being stimulated. Okay. The motor end plate on a muscle cell, a muscle fiber, has a much larger surface area with a much higher density of these receptors, these protein receptors with their channels, right? This allows for a much higher density of these receptors than we see in a neuron-to-neuron connection, okay? And basically, if you have a whole lot more receptors, you can get a whole lot more sodium ions flowing in, right? So that, the last point here is basically the result of these, what I've explained in the first 3 points, is that what this results in is that there are many more receptor channels in the muscle cell that open up in comparison to a neuron-to-neuron connection. So this results in a stronger graded potential in an EPP than for an EPSP of a neuron-to-neuron connection. Okay, and then just coming back to what I said already, a single EPP, a single end plate potential, is sufficient in most cases to initiate an action potential. Okay, so one action potential in a neuron triggers one action potential in a muscle cell. Okay, but the acetylcholine binding of these receptors in the postsynaptic membrane is a reversible process, and what happens is your acetylcholine binds very briefly to these receptors, these cholinogenic receptors, in the receiving muscle cell membrane. Okay, so it binds very quickly, and apparently it takes about 1/1,000,000th of a second. I don't know about you, but I struggle to think about dividing a second into 1,000,000 parts. But anyway, this is how quickly these acetylcholine molecules bind and detach immediately from these receptors. Okay, so it's very quick. Now, they bind, they open up a channel, and then they detach. Okay, remember that this acetylcholine can be inactivated or basically destroyed by acetylcholinesterase, the enzyme that destroys it, which is located in the motor end plate. Okay, now what I want to explain here is this whole process whereby basically if acetylcholine is removed from this whole process, from this motor end plate region, then you don't get any end plate potential. Okay, you do not get a graded potential happening in your receiving muscle cell. Okay, so you have to have this acetylcholine present and binding with these enzymes for greater potentials to happen. So what happens is, because this acetylcholine can bind so quickly and detach with the receptors, it can essentially, it can either, after binding with a receptor, it can either get destroyed by the acetylcholinesterase, okay, and then takes no further part, or it can rebind, it can reattach, very quickly can reattach to receptors if another action potential arrives, okay? So if, for instance, you get one action potential, causes the whole process we described of the neuromuscular synapse, and we end up getting acetylcholine binding with a protein, with a receptor. That will then be open up sodium channels and generally cause muscle contraction, via setting off the rest of the process. Okay, so the end plate potential. But if this now, then after this happens, the acetylcholine detaches itself from these proteins, and it can, if another action potential is received, it can very quickly reattach to a receptor and set off another muscle contraction. So if you're getting a very large amount of fast flow of action potentials arriving at this neuromuscular synapse, then there won't really be time for the acetylcholinesterase to destroy much of this acetylcholine substance, right? Because you're getting a rapid attachment, reattachment, attachment, and reattachment of these this neurotransmitter substance going on, and basically, like the acetylcholinesterase, okay, the ACHE. This substance basically determines whether a muscle fiber keeps contracting or relaxes, okay? Because the balance of this acetylcholinesterase, if on the whole acetylcholinesterase is killing off your neurotransmitter molecule, then you're going to get a relaxation of the end plate potential, okay? So you're going to get a return to resting potential, and you'll get a relaxation of the muscle fiber, and you won't have muscle contraction going on. If there's a lot of action potentials being received by this neuromuscular synapse, then you will get the constant release of more neurotransmitter substance from those vesicles, right, that escape from the axon. And as more and more of this neurotransmitter floods the motor end plate, you're just going to keep getting this EPP, okay, this end plate potential. You're just going to constantly be stimulating the muscle, which is necessary if you actually need to affect an action urgently in the muscle, okay? So the sort of the balance of how much of the acetylcholinesterase is present in this motor end plate region, balanced against the amount of the neurotransmitter substance, the acetylcholine. So the balance in the concentration of these two substances, okay, in this motor end plate region, is what's going to determine whether your muscle is basically contracting or relaxing, okay? So it's going to determine whether EPP is happening over and over again or not happening, okay? So just to finish off the lecture by looking at synapses and integration, and how do we integrate all these different synapses that are happening in the body. I just want to recap on the terminology quickly. So remember that EPSPs, okay, remember from the neuron to neuron connections we did in the previous lecture, EPSPs stand for excited excitatory postsynaptic potential. And what these are is basically a small depolarization that's caused in the postsynaptic neuron. So it's in a neuron-to-neuron connection, okay? By small depolarization, remember that's because you are setting up a graded potential here. So it's a small change in the polarity, small change in the small reversal of the charge. So that's EPSPs, okay? The IPSPs, inhibitory postsynaptic potential, it's the opposite, okay? It's a small hyperpolarization of the postsynaptic neuron. So a small, it increases the polarity, hyper to increase, remember? So hyperpolarization increases the negativity of the charge that we see at resting potential in a normal situation for a neuron cell, okay? So, so if a neuron cell is at resting potential at minus 70 millivolts, you're going to get, if a small hyperpolarization would be, for example, taking that to a state of minus 80 or minus 90 millivolts, okay? In other words, further away from threshold potential, further away from setting off an action potential. So in IPSPs are inhibitory, are called inhibitory because they lessen the chance of they lessen the chance of an action potential, okay? Excitatory postsynaptic potential are generally take the membrane potential towards a state of action potential, closer towards that, right? So a more positive state. Okay, so these two terms I just wanted to recap on. And remember, these are graded potentials. And they are produced, here's some more lovely terminology for you. These graded potentials are produced by excitatory presynaptic input, in the case of EPSPs, and inhibitory presynaptic input in the case of IPSPs, okay? It's kind of a fancy way of just saying that the EPSPs are generated by neurons, in other words, presynaptic input, so presynaptic neurons that are excitatory, that are causing excitation in receiving neuron, and the opposite way, vice versa, for your inhibitory presynaptic input, okay? So I want to talk about integration now, okay? So we've talked about individual electrical synapses. We've talked about individual chemical synapses, so chemical synapses between neurons. And we've talked about individual chemical synapses between neurons and muscles, okay? But in reality, there's thousands, hundreds of thousands, well, actually millions of these different connections going on, these different synapses happening all over animal bodies, okay? And all these, the EPSPs and the IPSPs, unlike for action potentials, okay, which are all or nothing events, these graded potentials, they can vary in magnitude. They also have no refractory period, okay? So they can happen in combination at the same time, and they can be summed, okay? So in other words, added on top of one another. So this ability of graded potentials to be summed turns out to be very important in our understanding of how synapses, whether they result in an overall excitation or inhibition of a receiving cell. Okay, so this summing of graded potentials, known as summation, you get two major types. You get temporal summation. Temporal means time related, so time. Temporal summation is is basically when you have repeated firing of a presynaptic neuron, and the graded potentials produced by these repeated firing events are summed in time. So they're so close together that they're actually causing a buildup of the graded potential just because they're happening so close together in time. Spatial summation is when your graded potentials, your EPSPs, they originate simultaneously from a bunch of different inputs, so different synapses, presynaptic inputs. Because remember, a neuron can have lots of different connections, right? And all these different synapses coming from different parts, from different parts of the cell. From different neurons, can now they all come together, right? And we sum them, and this is called spatial summation. Okay, so this is easier to explain with the diagram that I'll look at next. So just to point out that the excitatory postsynaptic potential are in sort of direct opposite polarity to the IPSP, to the inhibitory potentials, right? So these two types of potential cancel each other out, okay? So if you had an equal EPSP acting against an equal IPSP, the excitatory effect and the inhibitory effect would cancel each other out. In theory, they would cancel each other out perfectly, okay? Right, so to look at the overall, to sort of determine whether there's going to be an overall excitation or inhibition of a receiving cell, we look at a composite of all the EPSPs and all the IPSPs that are occurring. And this includes temporal summation and spatial summation. And the composite of all these results in what's known as the grand postsynaptic potential, okay? So the grand postsynaptic potential is the overall potential electrical charge that results from all these different synapses that are happening with with this postsynaptic cell, okay? So this that results in that is what we end up with, the grand postsynaptic potential, and that is either in the direction of stimulation, excitation, or inhibition, okay? So let's look at that with a diagram. So if we look here at an example where we have our, in this example, we have two excitatory inputs, okay? So these are two excitatory presynaptic inputs and one inhibitory presynaptic input. So this is a hypothetical example, okay? Just to try illustrate the process of summation. Two of these excitatory inputs come in, and they have a stimulatory effect. So excitatory, your inhibitory input will inhibit the receiving cell from action potential. Okay. So here we are talking about these inputs synapsing on your postsynaptic cell. Okay, so this is the cell body of your postsynaptic cell. In this case, a postsynaptic neuron. All right. So here we record the potential. Okay, so the membrane potential of the postsynaptic neuron. Okay. So this is a simplified situation, of course, but we want to look at basically the four main scenarios that can occur. We can have a scenario A where we have no summation. In other words, in this case, if an excitatory presynaptic input, this input here, for instance, is stimulated a second time after the first EPSP event, okay, so after the first stimulation, if it's stimulated a second time after the EPS, so after the EPSP in the postsynaptic cell has died off, then a second EPSP, so a second excitatory postsynaptic potential will happen, and it'll be of the same magnitude, okay, as the initial one. So in this case, you've got your basically in this case, you're not going to get any summation because the first process of stimulation is finished, and then you get another stimulation that is separated from the first one. So there's no, you know what I mean? There's no piggybacking, or there's no summation here. There's no overlap. Okay, so in scenario B, our temporal summation scenario, if our excitatory, let's look at EX1 here, okay? So if our excitatory input 1 is stimulated a second time before the first EPSP has died off, okay? This will add on to, okay? So in other words, this will sum with the first EPSP and will result in a temporal summation, okay? So this will result in summation of the potential, of the electrical potential, which may bring the postsynaptic cell to threshold, in other words, to plus 30 millivolts. So temporal summation can result in bringing, if you have these greater potentials overlapping in time, the result can be an action potential, okay? The third scenario C, is your spatial summation. So here, okay, we're looking at here spatial summation. So if you have simultaneous activation of EX1 and EX2, so if simultaneously these two excitatory presynaptic inputs are activated at the same time, then we call it spatial summation, because from different spatial areas of this postsynaptic cell, you're getting stimulation, okay? So this would result in depolarization, right, towards action potential, and this can result these greater potentials jumping, sort of adding together spatially into one larger signal can cause an action potential. The last scenario is your cancellation, okay? This is just simply when the inhibitory presynaptic input coming in here, if it happens at the same time as one of these excitatory inputs, then the two cancel each other out, right? Because if they are of the same overall charge, but in an opposite, they cause opposite effects, so inhibition and excitation, that will basically result in the membrane staying at resting potential, okay? So the balance of these spatial and temporal factors, you know, the balance of the spatial and temporal, you know, the number of connections going on and the number of connections that are inhibitory versus excitatory will determine your overall, you know, whether the postsynaptic cell records this as an action potential or not. Okay, so in other words, your grand, what do we call it? Your grand postsynaptic potential will be determined by the factors that we've explained with a simple scenario in this diagram. Okay, so make sure you understand how this works. I know it's quite finicky stuff, but it actually governed by some very, very simple processes, okay? Like basically excitatory effects canceling out inhibitory effects and vice versa. And yeah, temporal and spatial summation in theory are just simple adding together of inputs, okay, and resulting in an overall summation, which can be either cause an action potential or not result in a charge or a potential that is at -50, okay. In other words, not result in a strong enough depolarization to set off an action potential, okay? Just to remind you guys that, you know, we've been looking at this at synapses, and threshold potential is not uniform throughout the postsynaptic neuron, okay? So it turns out that the lowest threshold is present at the axon hillock, okay? So remember, the axon hillock is where the cell body of the postsynaptic cell neuron, where the cell body meets the axon. You have your axon hillock, and this region is where you have the lowest threshold. This is because it's loaded, it's packed with high density of voltage-gated sodium channel. Okay, so it's very sensitive to it's very sensitive to any sort of depolarization. Okay, so it's a highly sensitive region, and it has a lower threshold than what you would see in the dendrites or the cell body of a neuron, okay? The sensitive region is where you would set off your action potential, and that would, once that's initiated, nothing has to be added, right? It just goes along the rest of the axon, propagated to the end of the axon, okay? So we've talked about synapses and how electrical charge flows along the axon of one neuron, and then synapses chemically into the dendrites and cell body of another axon. So I just wanted to point out here that once this synapses happen, action potential then carries on along the next neuron, as long as an action potential is set off in the axon hillock zone of the neuron, okay? So then it carries on along the next neuron. And so the process is repeated, and this is how electricity flows from neuron to neuron throughout your body, okay? So just lastly, to point out that how enormously complex the nervous system is, and just how mind-bogglingly, crazily, like, I can't get my head around it, but how complicated this whole thing is. So one neuron can have thousands of other neurons synapsing on it, okay? So we call this convergence. So this is where a receiving neuron, a postsynaptic neuron, has thousands of presynaptic neuron terminals synapsing on the postsynaptic neuron, okay? So converging on. Convergence means to come together, right? Then we have divergence, which is when a presynaptic neuron, it branches, so it takes its signal and it branches this off in axon terminals. In other words, the signal diverges and synapses with many other cells in the body, okay? Other neuron cells or muscle cells. So this is divergence, right? So convergence can happen. A neuron can both receive thousands of signals via convergence, and it can give off thousands of signals via divergence. So if all these synapses are receiving and giving all these thousands of signals in just one sort of connection, you can imagine the complexity of all the different connections in animal bodies. So for instance, in the most complex case, we have the human brain, right, which has an estimated 100 billion neurons, okay? Now, because these are all synapsing with each other through convergence and divergence, this results in a number that has been estimated, I don't know how the hell they did this, but it's been estimated at 10 to the 14 synapses. So 100 quadrillion synapses in your brain going on probably right now. It's kind of crazy stuff. Yeah. So basically, I hope I've left you a little bit in awe of what's going on in your head. The wiring system, the electrical wiring in an animal, in most animals, or at least complex animals, such as vertebrates and higher forms of animal life. These wiring systems are way more sophisticated way more complex than any of the mega computers we have yet developed, right? So pretty amazing systems, these nervous systems. Just to leave you off with the picture, just showing the convergence situation, which is where we have our all our axon terminals converging on a cell body of a receiving neuron. And then divergence, which is when we have a single neuron here sorry, divergence is when we have our neuron here giving off its branching into different axon terminals, right? In this case, 4 are illustrated, but in reality, there can be like hundreds, thousands. So it diverges and connects with a whole bunch of other neurons, right? So it's like almost it's almost almost infinitesimal connections going on, because each of these single neurons connecting with a bunch of other neurons, and then they in turn connect with a bunch of other neurons. So this divergence is massive, okay? And then it leaves one in awe just thinking about the conversions. I mean, how many signals are being received by one neuron here from all these presynaptic input? And then, as I said, all these inputs are giving excitations and inhibitions, and they all result in a grand synaptic potential for this neuron, which decides whether an action potential ultimately is going to flow along this axon, from the axon hillock, and synapse with other neurons, or is it not going to happen, okay? So these constant electrical impulses are sort of on-off impulses that are firing around our bodies, okay? Okay, so thanks very much, guys, and hope you learned something today. I know it's a lot to take in. All the more reason to get some studying done, eh? Okay, thank you very much.